All right, well, it's indeed a great pleasure, and I'd very much like to thank Bengt for inviting me. I know Bengt primarily through this review journal that's been advertised several times, Quarterly Reviews of Biophysics. And I come from the biophysical side, but this is the first meeting I've been at where there's been such a superb interaction between chemistry and biophysics. So I think that's, for me, it's the highlight of the meeting. But I'll come back at the end and say a little bit more about Bengt. So yeah, and so I started out about 50 years ago in structural biology, and when I started as a student, there were only two protein structures. So it was a myoglobin and lysozyme, and a low-resolution map of hemoglobin. And now there are well over 100,000 sets of coordinates deposited, NMR, X-ray, and so on, in the protein data bank. So you could argue, you know, maybe it's in its decline now, but I don't think that's true, because all of the methods that have been developed have become more and more powerful, and so now you can do things that were unthinkable before. And so there are methods, and I'll tell you two or three of the ones that I've been involved with, which makes this a kind of golden era for structural biology. So you can basically do almost anything that you want to do. So if you're a young person starting out, rather than looking for another field, we would encourage you to, you know, carry on in this more biophysical thing. Okay, so I've picked these three topics to talk about. One is how I got involved with electron microscopy in the beginning, which I think has still a great potential that's not been fully realized. And then, having worked on bacteria rhodopsin for a few years, and found out that a whole family of 400 human genes that are all involved in physiology and so on, very important in medicine, the Gproteincoupled receptor family had some resemblance in terms of having 7 transmembran. We then went to work on this and found it was extremely difficult. So some methods were developed in Cambridge, particularly with Chris Tate, that I'll tell you about, that's been very successful, and it can be applied to anything, not only membrane proteins, Gproteincoupled, but any protein really that you're having difficulty with. This is an approach that's useful. And then finally, I'll tell you about what we've been doing for the last 10 or 15 years, which is to try to develop the single particle electron cryomicroscopy method so that you could get really good high resolution structures easily without crystals. So first, electron crystallography. So we started by working on this membrane fraction from Halobacterium halobium, as it was called originally. Walter Stekenius and his collaborators discovered these 2D crystals that are in the membrane of the bacterium, and when you isolate them, they can form beautiful little patches. They diffract as an almost perfect two-dimensional crystal. And so with Nigel Unwin, I made this transition from doing X-ray crystallography to doing early stage electron microscopy. And in 1975, a long time ago now, we got this very low resolution map of bacteria rhodopsin. At room temperature, no cryo, no detectors, it was all done on film and so on. And then over about the next 10 years or so, we worked at the various hurdles and barriers to the method, and gradually moved from this sort of 7 angstrom map to about 5 angstrom map. This was done by molecular replacement, trying to do molecular averaging and cycling, to eventually taking images of the 2D crystals in a cryomicroscope to get to high resolution. And finally this map in 1990, where you can see from these are 7 transmembrane helices. Protruding from the helix, you can see these side chains at higher resolution, and then with a model interpreting that map, like tyrosines, phenylalanines, tryptophans, we could end up with a complete 3D model of the structure using electron crystallography rather than X-ray crystallography. And then over the next 5 or 10 years, another group in Japan, Yoshi Fujiyoshi, made improvements in the microscopy and redid the structure, probably slightly better. And then after that, lots of X-ray groups eventually solved the problem of how to make really good diffracting 3D crystals. And at the time we made this slide, there were 6 coordinates. Now there are 100 or so in the database. But the other thing you can do by electron microscopy. When you look at the kinetics of the photocycle, this would be resting state of this colored protein which absorbs light and pumps protons. So it's a very inefficient form of energy conversion, photosynthesis if you like, maybe 5% efficient compared to the green light plant photosynthesis that you heard about from Jim Barber, which is much more efficient in overall terms. There's a series of intermediates, but the big structural change occurs at the bottom here, where a proton is released on one side of the membrane by structural changes from the chromophore retinal, and then taken up from the other side of the membrane. So there obviously has to be a ratcheting for opening to one side and then opening to the other side. So the idea which you can do with these thin 2D crystals is you have this environmental chamber with a pair of forceps, and this is the electron microscope grid, 3 millimeters in diameter. You put a drop of these crystals, maybe 100,000 crystals on the grid, and then you fire it down here at a few meters per second into liquid ethane to trap it at the temperature of liquid nitrogen temperature. So the whatever intermediate you've created by the flash of light at the beginning of that photocycle, you then trap it. You can do it at half a millisecond, 1 millisecond, 10 milliseconds, and so on. And this is exactly the same technology that's used now in single particle EM or in tomography. So it's a general approach to specimen preparation. So when you do that, you can collect data and get a difference map, just like you would if you were doing a change in structure by other methods, positive contours, negative contours, showing a movement of about 3 or 4 angstroms of 2 of these transmembrane helices. So helix 6 and 7, 7 being the helix to which the retinal is covalently attached by the lysine. So the proton is released on the outside of the cell at the bottom when you flash it with light, and then these 2 helices move apart, opening up the channel to the inside of the cell for it to be reprotonated. So we felt we had a good idea of the structure. And actually this quite large structural change hasn't been clearly seen in any of the subsequent X-ray crystallographic structures, because the 3D crystals, which need to be very well ordered, trap the protein in the resting state, and then that conformational change is prohibited. And then in the family, this is a light-driven proton pump, but there's a range of other functions of homologous proteins. There are light-driven chloride pumps that are homologous, and signaling proteins that do both phototactic attraction and phototactic repulsion. And this same structural change is then coupled to a range of physiological responses and energy transduction responses in the cell. And there are now thousands of these sequences from a variety of not only bacteria, but also higher organisms with a range of functions. And also there's the light-activated channel opsin that's also been used as a tool in photoactive genetic linkage for stimulating nerve impulses in nerve cells. So it's both a natural phenomenon with a range of functions and a tool in modern nanobiology. So then I was going to switch to this the second topic, which is this very big and very important family of G-protein-coupled receptors. And after we had that structure of bacteriodopsin, my original thought was, well, you know, that's an era finished. We should move on and do other things, motility, actin, tubulin, and so on. But this, first with rhodopsin, and then the beta-adrenergic, and then a whole range of these G-protein, it became apparent this was a very important family. And since we are supported by the Medical Research Council, we had felt a certain obligation to do this. So we started, and this was with Gebhard Schertler, who's now at the Paul Scherrer Institute in Switzerland, and a little bit later Chris Tate came, and for 15 years we had various grants we couldn't get them from the typical funding agencies, but pharmaceutical companies, and in particular Malcolm Weir in GlaxoSmithKline. He felt this was a very important area, and we were supported for about 10 years with between 2 and 4 people to work on different G-protein coupled receptors. And however hard we tried, we could never get crystals, not 3D crystals, not 2D crystals. So maybe 30 man years, many papers were published, people's careers were not ruined, they got good jobs, but we never got a single crystal. But these were the three, and so eventually in 2005, we managed to get a fourth grant from a different pharmaceutical company, Pfizer, with some backing from MRC Technology, which is a sort of development gap funding to try and bridge the gap between academic blue skies ideas and then practical value of them. Anyway, we picked out of this whole Gproteincoupled, this is family A of GPCRs, there's also B, C, and so on, but and they're related, but and I think Kurt Wuttrich showed this in terms of the protein structure initiative that had been done at Scripps, in which they picked different structures, and their approach was a bit different. They picked hundreds of these and then tried to express them, and then took whichever ones looked like they would work, and then there were lots of tricks like T4 fusion, cubic lipidic phase, and so on. But our approach was different. We picked a peptide, this was the Turkey erythrocyte adrenergic, Elliot Ross gave it. We picked these structures and we said we're not going to deviate, we want to solve these structures, and then if it turned out to be difficult, perhaps the methods could be used for all of the other ones. And after about 15 years, without any success, Chris Tate and I had heard a talk by James Bowie from UCLA, and he had been working on bacterial membrane proteins, and he'd shown that by making mutants, you could keep the function, or at least many aspects of it, and by making mutants, you could make the protein more stable. So the idea was to, and these were the four people who were involved with funding from Pfizer and MRCT, and they worked for 2 or 3 years each. And I'll begin with Maria Serrano Vega, who picked this beta-1 adrenergic receptor. And the idea was to make, as James Bowie had done, a completely systematic genetic screen where you express and you measure the stability by heating it for half an hour at, say, 30 degrees or 40 degrees. And so she made mutants, probably about 500 single-point mutants, originally by hand, but now, of course, you can do it with a multi-well plate with a robot. It's much quicker. So this was the sequence of the beta-adrenergic receptor. These were some of the mutations. This was the expression level. This is how much more or less stable each of these mutations were compared to the wild type. And originally it was just alanine. But in a second round, each of the amino acids, for example, glycine 67, Maria changed it to alanine, valine, leucine, and isoleucine, and then looked compared to wild type and picked whichever mutation for each of these amino acids gave you the biggest increase in stability. And then so this was in a second round where those amino acids residues which appeared to affect the stability. Of course, most of them killed the activity and were less stable or were neutral and had no effect, but a few of them, about 10%, gave you an increase in stability. And this is an approach that you can use not only for G-protein-coupled receptors or membrane, you can use it in principle for anything, spliceosomes, RISC complexes, nucleosomes, and so on. So then in a third round, Maria then combined the best mutations, and in the end she picked 4. So these are the gradually improved inclusion of different mutations. Here is the stability, the percentage of activity left in the wild type when incubated for half an hour at 32 degrees centigrade. And you can see you go to higher temperature, there's 1% left, and then there's nothing. And then as you introduce these mutations, you can see that it gets more stable, 5 degrees, 10 degrees, 15 degrees, and eventually this one, which had 4 of these thermostabilizing mutations included, was stable at 52 degrees centigrade, but 20 degrees centigrade more than the wild type. And this converts, if you keep it at 1 temperature, it's a factor of 300. And then later on, a student whose work I won't mention, Jenny Miller, carried on this, and it turns out if you carry on doing this, you can make them as stable as you want. She made it about 35 degrees centigrade, 5,000 times more stable. And then, you could continue if you wanted. So basically this general idea, systematic mutagenesis, careful combination, you have to have an assay so that you preserve the activity that you're interested in. In this case, it was ligand binding, and originally it was antagonist binding, but you can use an agonist and get two different states for the structure. So we think it's a general approach which, if you're stuck because your protein is not stable enough for NMR, not stable enough for crystallization, not stable enough perhaps for single particle EM because of the freezing your specimen in at an air water, you can then use this approach. So when this protein was then taken by Tony Warren and set up using exactly the same crystallization screens that had completely failed, my slides, that was got out. Here is a crystal. So he got a lot of different crystals in the first screen and managed to get this 3 angstrom, 2.5 angstrom structure. And in the end, all three of them were solved. First one by Maria, beta-adrenergic, then adenosine A2A in both agonist and antagonist forms. And then most recently, Reinhard Grieshammer, who had started out this work on the neurotensin, the peptide receptor. This one turned out to be the most difficult. And they needed to have 6 mutations. They needed to do some of the other tricks that had been introduced by, for example, Brian Kubilka had this T4 lysozyme fusion, and cubic lipidic phase crystallization. So eventually, after about 20 years, many papers were published, different angles on it, but the structures of all three of them were obtained. And so then we thought, okay, that's an academic lab, 3 structures, but Chris Tate and I got involved in starting a little company called Heptares, and they've now industrialized this, which was quite labor-intensive initially, and they've now solved a dozen or 20 GPCR structures that are valuable drug targets, and now they have some drugs in clinical trials. Okay, so then let me come to this last topic. So alongside this, the idea was, well, maybe even if you make your proteins very stable, it would be very nice not to have to bother to crystallize them. And it turned out originally the idea of 2D crystals was maybe it would be easier to make 2D than 3D, but it turns out not. You just as frequently get a disordered 2D crystal as a disordered 3D crystal. So a lot of the proteins people have worked on, sometimes for many years, they try this, they try that, they get a crystal, it looks beautiful, they diffract it, and it diffracts to 10 angstroms. They carry on for another few years, it diffracts to 7 angstroms, and eventually they sometimes have to give up. So there are a lot of structures. So the idea was, what about single particle cryo-EM? So I'll just give you a brief introduction to that. So here was a slide that Tim Baker and I put together for a review in the International Tables for Crystallography about 10 or 12 years ago, just to explain to people about what is single particle electron cryomicroscopy. And the idea is you make a grid and plunge it into liquid ethane. This is a method that Jacques Dubochet introduced in 1982 at EMBL. And these are 4 different specimens. So the method is the same, and you can apply it for a whole range of specimens. So this was a field of view of some E. coli 70S ribosomes from the work of Joachim Frank, published in the year 2000, and it was an 11 angstrom map, state-of-the-art technology then. Here are the 70S ribosome particles. This is the ice in between them, and the idea is you digitize this pattern, put it into a computer, and then try to work out what angle of view each of these ribosome particles are. And then once you've got the angle, you put it together and you calculate the 3D map. And they had this 11 angstrom map, small ribosomal, large ribosomal subunit, tRNA, and one of the long RNA helices. But you can also apply the same method to particles with symmetry, an icosahedral particle. This was at about 7 angstroms in 1990. Helices, actin decorated with myosin, tropomyosin, different domains of the... And then this was a different 2D crystal from the one that we'd worked on before. But this was light harvesting complex from green plants from the work of Werner Kuhlbrandt. So this gave an impression of the range of specimens you can use. You can also... Further over to the right, in principle, you can do tomography of the same cryo-EM specimens, collect data dose fractionated in different views, and then without having a repeating structure, you can get a 3D tomogram of bits of your cell or subcellular fractions and so on. But because of radiation damage, the resolution you can get if you only have one copy of something, for example, part of a cell machinery and so on, it's limited to about 20 angstroms, we think. In practice, it's more like 30 or 40 angstroms at the moment, but it will get better. Whereas if you can average images of the same molecule in a 2D crystal or a helix or single particles in which you can identify the orientation, then in principle you can get the structure at any resolution you want. In practice, 10, 15 years ago, the resolution was limited. But the thing that's made the big difference recently are the advent of these new detectors, and we've worked on this as well as other people. This is what we've been using in all the previous work. This is film, and this is the resolution. This is the sort of Nyquist resolution limit of any detector, which is 2 times the pixel size, and this is 100% of the detector. This means when you've detected every electron or X-ray, if it's an X-ray detector, perfectly, you'd get a 100% DQE, and if it went over here at 1, that would mean not only did you find where all of them that they exist, but you'd also find accurately where they are. So the fact that these curves are not 100% means you've missed some of the electrons, and also that you haven't identified where they are. But each of these 3 colored curves are the new detectors that have been available for about 2 years now from Gatan, FEI, and a company in San Diego called Direct Electron. All of them are better than film, and they have the added advantage that they come out as movies. So you can do post-processing, you can throw away frames, you can give them weighting and so on. And then recently, so these were the 2 we've been involved with. There are some, and we hope, since film at 30% was what we were doing, and the big improvements recently have come from these detectors, which is more or less doubling the DQE. If you can get it up to 100%, we think the methodology will be really valuable. So what I'd like to do is just show you 2 membrane proteins, but first a couple of other structures that we've used. One of them as our benchmark, and then one of them because ribosomes have been another structure that's been used to measure progress. So this is a holy carbon. This is carbon film, this is ice, and this is a suspension of the enzyme Betagalactosidase, which we picked as our test object about 1997 and so on. This picture in the background was taken in 1997, and this one in the middle here was one of the new detectors. And actually, if you look at it carefully, these ones, you can see the particles, but there's not quite enough information to be convinced that you can see all the different views. Whereas in this one, with a bit of experience, you can say, yes, this is a top view, side view, 45-degree view. And this was our efforts with different computer software. We didn't believe any of these until we got to this 2011, which was actually done on film, but with some new tests that we were able to do to prove to ourselves that it was correct. And then with the new detectors, we got better resolution. And then with a new program, Relion, that had introduced maximum likelihood methods for identifying the particle orientations and for calculating the maps, there was a further improvement. And this is about a 3.8 or 4 angstrom map from the same data that Georges Cherez, who wrote this program, did about a year and a half ago. And it now shows beta sheets, which are 4.8 angstroms apart, as well as alpha helical stretches of the protein. And then another structure, and that's where somehow or other that slide got out of sequence. This is a field of view of mitochondrial ribosomes with one of these 300 kV microscopes and one of the new detectors from work done by Alexei Amunts, who's in Venki Ramakrishnan's group at MRC LMB. And Alexei has just agreed to become, or he's just been recruited to come to Sweden at the end of this year and help to set up hopefully productive cryoem group here. But they worked on this mitochondrial ribosome, and mitochondria have very few ribosomes, so when you purify them, there's always a bit of cytoplasmic ribosomes mixed in, maybe even 50%, and there are some of the non-intact, let's say 54S rather than 74S. So what is done using this Shor-Scherer's Reliant program, you can not only find the orientations of the particles, but classify them so that you can then subsequently in the computer figure out which of these images refer to mitochondrial ribosomes and which ones refer to cytoplasmic ones, and then put them into separate classes and get the structure of all of them from one data set. And the ones that they were aiming for were the mitochondrial map. So they averaged these 4 classes and got these maps with the large and the small subunit, with the red ones being subunits that hadn't been seen before in any previous ribosome. And to show you quality of the map, we have to go back now to where yeah, so here would be a little stretch of RNA Watson-Crick double helix with the base pairs resolved, because it's a 3.1 angstrom map. They're very well resolved, and then with a top view you see the Watson-Crick and the pairing, and then with a side view a little bit of an alpha helix, a beta structure with a loop, and then even they believe one magnesium ion at one of the loops. So this is one of the really good quality maps. And then the 2 membrane proteins that I would like to show you are this complex, mitochondrial complex 1, which Judy Hurst and Jai-Pen Ju have worked on for many years, maybe 10 years, and before that John Walker's group had worked on it. And their goal had been to purify it, and they did have crystals, but they never diffracted very well. And so they brought this specimen over about a year ago, and Vinoth Kumar took a few pictures, and this is one of the pictures, and you can see it's a 900 kilodalton complex with 44 different protein subunits. So he picked perhaps 40,000 of these particles, and then first did classification in 2D, looking for patterns. And I think you can see here, for example, this side view here would be a cytoplasmic domain, this would be the membrane domain, and there are even striations here, which are some of a sort of first view of the transmembrane helices. When these are then all put into 3D, they got a map which was published about 6 or 9 months ago, showing the entire structure at about 5 angstrom resolution. And when contoured at a high contour level, you can see these 8 iron-sulfur complexes which conduct the electrons from NADH down somehow or other into the membrane with a conformational change and then proton pumping across the membrane. And then in the membrane section, there are 78 of these transmembrane helices. So they're individually resolved, and currently they're now working, of course, to improve the resolution. If you drop the contour level to a 3rd lower level, then you see this band of detergent that's used when you solubilize, remove the membrane complex, and then purify it. And then prior to this work, another group, also in that same MRC mitochondrial biology unit, next to our lab in Cambridge, Leo Sasanoff had purified and crystallized a thermophilic bacterial version of this complex 1 involved in energy transduction, which is about half the size. So they were able to interpret this density using that mitochondrial complex. And so the pale colors are the subunits that are homologous between the bacterial and the mammalian. This would be bovine mitochondrial. And then the dark red subunits are the new ones, supernumerary subunits, if you like, that are involved in the mammalian, the mitochondrial complex 1 rather than the bacterial one. And they were able to identify about 14 of them, so that they have, I think, 28 subunits out of the 44 assigned and identified. But now with higher resolution, they should be able to do better. And then I'm going to finish by showing one other membrane protein structure, which is from the work of Xiaochen Bai and Shor-Scherers in Cambridge, and then Liu and Yigong Shi in Tsinghua, who have been working on this gamma secretase, and many other groups around the world have been working also on this enzyme. It's a small membrane-bound enzyme, about 170 kilodaltons. There are quite a lot of papers published with some preliminary sort of negative stain work, 20 angstrom resolution. No one's ever got a crystal that I know of. A lot of people have tried. This was also published 6 or 9 months ago. It's a small protein, and nominally the resolution is only about 4.5 angstroms. It allowed them to identify these 19 transmembrane helices, and then the enzymatic domain on one side of it with the cross-section here in that blue rectangle indicated here, and then parts of the structure as beta structures which you need about 4.5 angstroms to resolve, and alpha helices with the beginnings of side chains. So both of these structures, both the complex 1 and the gamma secretase, are published about 6 or 9 months ago, but both of them have now been taken basically just by doing more work with the same specimen, the same microscopes, the same computers, to 3.5 or 3.8 angstrom resolution. So really it does work quite well. So I'd just like to finish then just by acknowledging the work, some of the people who I've worked with and others that are at LMB. So the original bacteriopsin work. The person I didn't show a slide of is Guillaume Le Bon, who took over from Francesca Magnani, and alongside work on antagonist-bound adenosine A2A Gproteincoupled receptor, he did an agonist-bound, which is the one that I showed, which, because it's a different conformation, the ligand binds, you open up, you signal to G proteins. It involved a completely separate set of mutations. So this conformational thermostabilization idea means you can focus not just on a structure, but a particular conformation of that structure, picking a certain number of mutations to stabilize that one structure, essentially trapping it in that structure, and then making it suitable for 2D or 3D crystallization or single particle cryo-EM. And then in the single particle EM, I mentioned Alexei Amuts, who's coming to Sweden, and then this group led by Georges Cherez in Cambridge and Yigong Shi in Tsinghua in China. And then I want to finish, so that's the end of the talk, by coming back to Bengt and mention a journal that goes close to all of our hearts. This is QRB, and here are the current editors led by Bengt. And here is the profile of citations of quarter reviews of biophysics. It started in 1968 with Arne Engström from Sweden was the founding editor of QRB, and he worked hard and built it up. And you'll notice that these curves, they go up and down, and we think that the slope as well as the absolute value of the citations is an indication of the quality of the editorship. And you'll notice that particularly here at the end, which is the Bengt Nordin era, it's doing particularly well. And it seems to, in fact, it's remarkable that it's a sort of linear thing. That doesn't happen with every journal, I should say. I wasn't going to mention that. Before me and after me, it wasn't, there was a sort of plateau period. But anyway, the point is that we all wish you happy birthday, Bengt. We hope you will carry on, not only with the main journal, Quarterly Reviews of Biophysics, but the new journal, QRB Discoveries, from all the authors, editors, readers, and publishers. So Bengt, thank you very much.
Thank you very much, Richard, for a fantastic talk. It's impressive what can be done today. So we have time for a couple of questions here before we start the panel discussion.
Very beautiful talk. Are there any general lessons you learned by the thermal stabilization of proteins? Were there any particular lessons out of the couples that you made that you could share with us?
Yeah, I can share the fact that when we asked for money in the beginning, we said we only were going to do 2 GPCRs. We said 2 is enough. And we said we would find general principles, which I think is what you're asking.
Yeah, that's what I'm asking.
And the people giving us the money who wanted to patent something said you cannot patent a pattern from 2, and that's why we decided to do 3. However, we then went on, and if there is a pattern, it's very, very weak. But there are, and one thing you can say is that residues that point outwards into what would be, say, aqueous solution if it's a soluble protein, or into the lipid part, as long as you don't make, an aspartic acid or an arginine point out to the lipid phase, it doesn't make so much. They're all the ones that are involved in the interactions within the protein.
Yeah, right.
But because they're all different, they bind different ligands, they're all ever so slightly different. So you basically, so our view is now you should basically just set up a semi-robotic system and do them all. In fact, what would be great, we could only do 500. If you have a protein of, say, 600 amino acids, you'd be better to do 12,000 mutations. That would be all of the amino acids in all of the positions. And then just have a robot do it. But you need a screen that's easy to do, and this was all done by radioligand binding, but in 96 well plates. So you'd be cloning, expressing, assaying without purification with a ligand binding assay. So you do have to think of the assay, but there isn't really any particular pattern.
I ask that because there are various computational groups who've been exploring this and have come up with some algorithms and things about stabilization, and it's also been found that in several proteins that these cation-pi interactions that you can introduce as mutations inside proteins dramatically stabilize folds. And I wonder if you looked into any of that.
Indeed, actually, there are a lot of groups that are very keen, just the same groups that do drug design in the pharmaceutical industry, they do protein folding, and I think we've had quite a few talks about protein folding. And actually, they are getting better and better, and Michael Levitt was saying we need better energy functions. I think we all would love it and realize that one day, these computers, you won't need any experiments. You'll just... particularly if you've got 100,000 or 1 million structures in the database, you won't need it. However, my intuition is...
It's going to be better to do robots now than theoreticians.
Yeah, but I'm afraid the experimentalists will have discovered all the structures before the theoreticians can calculate them.
Thank you. Thank you
Sorry.
very much.
Astrid?
I think it's on here. So the single molecule EM that you talked about last, I suppose you could try out your GPCRs also with that and maybe compare. What would you expect to be the difference between a mutated and a non-mutated GPCR? Would you see the same sort of thing?
Yeah. So what's the difference between the mutated and... So the mutated ones are locking it in one state. So... I think what you'd expect, of course, at the moment, it's quite, this single particle EM, there is a problem as you get smaller and smaller. Each image of each particle has less information, and there isn't enough to orient it properly. But with some technical improvements, we think we will be able to go down to 50 or 100 kilodaltons. Certainly, there are some of the GPCR family, for example, family C, a lot of them are in the form of dimers. So these are actually probably doable now. So there are, I know, other people as well as ourselves quite interested in doing that. But in terms of mutations versus non-mutations, I think the wild type will differ in that there will be kinetic barriers that are lower between the different states, whereas the mutations, you've locked it in one state. But we think that if you lock it in an agonist or an antagonist state properly, then because you've been assaying it all along to make sure all the ligands bind with the same affinity, so it ought to be very, very similar. So we're not worried about the fact, which some people object to, is to say mutations. You make 4 mutations, it must be a different structure. We think it's so similar, you can certainly use it for drug design. Without worrying about subtle changes like that. Yeah. Well, I think you'd still be better because they're small, you'd still be better to make two specimens, one with agonist and one with antagonist, rather than you can, in principle, try to sort them out in the computer later. But that works better with bigger things because there's more signal. The smaller ones, think you'd probably get a bit of a fuzziness still.
So we take one more question before the panel discussion.
When you continue to improve the resolution in your single particle electron microscopy, can you eventually take a movie of how ribosome make its proteins?
Yes. I
Yes?
think so, yes.
How long have to wait?
Well, no, you still have to freeze them. You still have to freeze them because if you, for example, we had a really nice talk with the graphene layers with water and nanoparticles jiggling around. You don't really want them to move during the exposure. So we think it's, and also when you freeze them, the radiation damaged fragments, they are released. And at liquid nitrogen temperature, only the big fragments, so for example, methane, ethane, nitrogen, as you're irradiating it, those are being given off as a gas. So you're left with a kind of core. So you have to stop them moving so that the images are not blurred during exposure. And then you get an additional advantage from freezing in that the radiation product of some damage resembles the original thing, so you get more data. And actually, there would be an advantage in the longer term also in liquid helium, where even smaller fragments don't come out. But at the moment, you get stresses and there's a so there are technical improvements coming that are going to make this really even better than it is at the moment. So actually, this is a really, that's why one of the reasons I said it's a golden era. So if you've got trouble with your stability, you can make mutants. If you've got trouble with crystallization, you can try. So there's so many things you can try now that it's really a lot of fun.
Great. That was a good conclusion of that fantastic